All right, thanks, Luke. So I'm Dan Ricciuto from Oak Ridge National Lab. I'll be talking today about some of the work we've been doing in the ELM, the E3SM land model, to develop surrogate modeling techniques aimed at helping us do uncertainty quantification within that model. So I'll be giving approximately the first half of the talk, and then I'll hand it over to Khachik. I'd like to also acknowledge Peter Thornton and Dan Liu, who've also contributed content and ideas to this presentation. Next slide. So I wanted to start just with a kind of a brief overview and motivation for why we're doing this kind of work within the land surface modeling community. And so when you look at uncertainty as it pertains to the land carbon cycle, most of the uncertainty that we see in papers comes from these multi-model ensembles from different modeling groups. And what we often see is very large spread in the model outputs. And on the upper right is an example of the cumulative land carbon flux from 1860 up through 2100. And you can see within the latter part of this century, the models diverge quite a bit, so that you can either have a sink or a source of carbon by the end of the century, depending on the particular model. And so that's just, of course, one of the quantities of interest that we were looking at in the outputs of these models. But there's little so far in the way of formal kinds of uncertainty quantification, at least at these kinds of global scales. And the reasons for that, of course, is that it's very expensive to run coupled Earth system models, but also even to run kind of offline land model simulations. And there's high dimensionality, meaning that there's a lot of uncertain parameters and inputs that go into these models, which in general, the more uncertain parameters that we have, the larger the ensembles have to be in order to quantify their uncertainties. And so some of the particular challenges that we have is trying to determine what processes drive the uncertainty in the outputs. And so on the lower right there is just an example of an ILAM diagnostic that's latent heat flux and the bias patterns that we see in ELM compared to observations. Also, what accounts for key differences among models, something that's kind of difficult to determine when we have different models from different groups using different model structures and different parameters. So we're also interested in determining whether model calibration can help us reduce uncertainty. And so a more kind of formal process to integrate models and data together. Next slide. So again, the case for running large ensembles, I think it's needed to understand how uncertainty in the model input parameters affects model outputs of interest. And as I already mentioned, there's a quite high dimensionality. I think there's over 100 parameters in the parameter file input that goes into ELM. But if we have an ensemble, we can use that as a basis to construct surrogate models, which enable uncertainty quantification methods like sensitivity analysis and parameter calibration, which we'll talk about in more detail later on. And so up until now, we've done quite a bit of work at sort of the single grid cell or single column level, where sort of one nice property of the land model is that we can isolate these single grid cells and run them since there's no communication currently between cells in the land model. So we use an MPI serial version of the model and develop a sort of parallel capability to run ensembles with lots of different parameter values. And we've done that with the default ELM. We're doing it now with ELM fates and the crop model. So at the global scale, we obviously there's a higher computational cost, so we can't run ensembles quite as large, but we can still run on the order of hundreds generally at low resolution. Next slide. So here's an example of that at the global scale. We've developed an ensemble of ELM simulations focusing on, in this particular case, on gross primary productivity. And so that's basically a photosynthetic input, a primary input for the land carbon cycle, but there's also a strong coupling between GPP and transpiration. And so we picked 10 parameters listed below there, and we constructed an ensemble of 275 members at approximately 2-degree resolution. And this is using the satellite to phenology version of ELM. So with the biogeochemistry turned off in this case, but we still get, the model still predicts GPP and energy fluxes in that configuration. And so these parameters are primarily focused on things that control photosynthesis, for example, the fraction of leaf nitrogen and rubisco being one of the most important parameters for that, but also parameters that control stomatal conductance, parameters that control the shape and orientation of leaves, and rooting depth, among other things, and temperature sensitivity types of parameters. So on the next slide, this just shows of those 275 ensemble members, if we start to look at some of the statistics of that. So on the top, we have the mean GPP, looping in a video from approximately 15 years, and so each frame represents 1 month. And so we see sort of the seasonal cycle of GPP appearing. We're in the northern high latitudes. We see higher values in the summer when the vegetation is active. And then if you look at the tropics, it's more, you see a more consistent pattern where there's always some activity, but you can see where the kind of the dry seasons are when things go lower, and the wet seasons when things go a little higher. And on the bottom is the standard deviation. So across those 275 ensemble members, basically what's the standard deviation of GPP. It's on a bit different scale there, but you can see that is basically proportional to the GPP itself, but it's some indicator of the uncertainty that we see as a result of those parameters. Next slide. So as I mentioned, one of our goals for creating these ensembles is to use that then to create a surrogate model. So what's a surrogate model? So what we define that is basically it's a good enough approximation of the full model, the full ELM, over the range of parameter variability that we're interested in. And so there are a lot of different names for these types of approaches. You might know them as meta models or response surfaces or emulators. But the point is basically it's a black box that takes in the same uncertain input parameters that the full ELM takes, and also the forcings that we're interested in. And what we get out of that is then a quantity of interest. For example, it could be global scale GPP, it could be GPP at a single grid cell, whatever we happen to be interested in and can get from the full model. And then we take that and use that for things like parameter estimation or optimization. It can be used also for experimental design, looking at where the uncertainties are high, or for forward uncertainty propagation. So on the next slide, we can see that when we start building these surrogate models, we're kind of hit twice by the curse of dimensionality. And so the first challenge there on the left is, as I mentioned before, we have a large number of uncertain parameters that go into ELM. And so one solution to that is to start with that high dimensional problem and then perform sensitivity analysis to pick the most important parameters and then move forward with those for calibration or other things. And so the sensitivity analysis tends to be a little cheaper in terms of the number of ensemble members that we need to get a good result. The other problem, though, is that we have a high dimensional output. So when we start looking at these global scales, it's not just a single grid cell. We have many thousands of land grid cells, and also in this case monthly outputs. And so in this case, where we're just looking at 2-degree simulations over 15 years, that's hundreds of thousands of outputs that we're interested in. And so something that Khachik will talk about is dimension reduction approaches that we can use in order to reduce the high dimensionality of these outputs. Yeah, you can move on to the next slide. And so first, global sensitivity analysis, which I mentioned, is a tool that we can use to enable parameter selections when we have a very high number of parameters to sort of scale that down for the next step. And so sensitivity analysis tells us basically how much output variance would reduce if a given parameter is fixed to its nominal value. We can also look at joint sensitivity. So in some cases, a quantity of interest might be sensitive to a combination of two or more particular parameters. So next slide. So we did that first with just the globally average GPP. So we took those ensemble members and we just calculated the global total GPP, and we can see sort of the order kind of the sensitivity. And so this FLNR parameter here on the upper left tends to be by far the most sensitive parameter, but there are a number of parameters which are sensitive, including some of the model slope parameters and others. So on the next slide, I think this is where, yeah, I'm going to hand it over to Khachik, and I'll talk a little bit more about the calibration and the dimension reduction approaches.
I just want to highlight how we construct surrogate in this spatio-temporal case, where we have high dimensional, basically a large number of grid cells and time units. And what we do is we build the Karhunen-Loève expansion first. This is essentially like a continuous version of principal component analysis. And then instead of building surrogate of each cell and each time individually, we build surrogates for polynomial surrogates for each of these principal components. So that's sort of the second equation. And we get this overall spatio-temporal parameter-dependent nice form of a surrogate that is really a fraction of a cost of a true ELM with some associated error addition to it, essentially. So what in a bit more details, let's think about there are like 3,100 land cells over 180 months of a simulation. That's 500K outputs for just GPP only, and we don't want to build surrogate for each of them individually, right? So instead of that, we build about 2,000 surrogate, one for each eigen or principal component, if you wish. And end result is a single spatio-temporal surrogate, which is about 10% relative error compared to the true ELM. And there is room to improve it. There are lots of other techniques that we can apply here, either increase the orders of KL and PC expansions, or use heavy machinery like neural network autoencoders to perform some of this dimensionality reduction. But the core point is that we are working in a reduced dimensional space first to develop the overall big surrogate. And the demonstration of two samples, basically, red is the true ELM and blue is KLPC surrogate, and how it mimics the behavior of the true ELM. You can see it here. One sort of side product of these polynomial expansion type surrogates is that we get sensitivity information for free because of the orthogonality. Basically, computing variance decomposition is analytically available for these guys, and we get essentially free information out of the surrogate about sensitivities. For example, the Harvard side and the Montana side, you can see the most important parameter is FLNR, and the second one is usually across all the sites that we have looked at is this MBB-opt parameter that I'll talk about in a little later. For example, in the Montana side, you can see the higher water limitation leads to an increased importance of stomatal parameters, just the one like MBP-opt, and that regulates transpiration. Now, this is a slice, a time slice at a given location. Now let's look at this sensitivity slicing in a different way. Basically, we're looking at time evolution over the whole grid. And you can notice that the scale sensitivity goes all the way almost up to 1, the color map, because FLNR is the most important parameter. And it is high in regions, pretty much in all regions where there is active vegetation. This is the fraction of leaf nitrogen in Rubisco. And the other one, the second best parameter is the stomatal slope parameter, MBBO, that I mentioned. And that one is more important over dry and cold regions typically. You can see from the video, let me try to play it one more time. All right, now having this surrogate, we want to constrain parameter space from these 10-dimensional large ranges to distributions of the parameters that actually match the observational data better. And for that, we're going to use this FLUXNET and FLUXCOM framework, the data that comes from there. FLUXNET is towers that measure CO2 and water energy fluxes, and FLUXCOM is a gridded, in this case, GPP benchmark, upscaled from this network using various techniques. And in this study, we are taking 96 high-quality sites selected for calibration. In fact, some of the results will be one calibration per site, or will be calibration using sites on the Western Hemisphere. And this is the visualization of the FLUXCOM data. This is the same type of video that you see before for the model, but this is the observational data. We kept the color map the same up to 15. You can see it never gets red. Really, the observational data is below the model mean. Model is over predicting typically compared to the observational data. And this next slide also kind of shows, again, another slice of the same data set. This is 2 different sites in US, and we are looking at time series of observational data on top of the 275 ensemble members. And generally, we see that ensemble members actually the mean over predicts what observational data provides. Now, how we can make use of this observational data to inform the uncertainties and perhaps reduce the uncertainties. Our approach to go is the Bayesian approach for basically parameter calibration. It is a very flexible framework, of course, with allowing incorporation of various sources of uncertainty. And typically, we do Markov chain Monte Carlo to build parameter distributions on the input that match the best to the observational data. But Markov chain Monte Carlo has its challenges. It's high dimensional, quite ugly PDFs, but there are advanced methods available. The most important challenge, though, is that every time we march in the parameter space to try, we cannot afford to run online the model and then compare to the data to compute the Bayesian likelihood, etc. So this is why we needed the surrogate to replace the model and run this calibration online with the surrogate. After the fact, when we get this calibration done, we can do prediction, and then we can decompose the variance on the prediction into various pieces, including parameter contributions like global sensitivity analysis that Dan mentioned, also surrogate error, data noise, and one little piece that is essentially state-of-the-art work that we have done recently is model structural error also is a component of this prediction variance that we are building into this Bayesian likelihood. I'll spare the details of that, but in principle, that embedded model error approach allows us to augment this extra term in predictive variance that is not very classical, let's say. The overall workflow looks like this. Essentially, we build a surrogate, we embed structural error components in it, and then the calibration mechanism uses this embedded surrogate to get to the posterior of parameters. After that, we can do predictions, posterior predictions. So I'll be using this language, prior prediction and posterior prediction. Posterior is the one after seeing the data, after doing Bayesian calibration. So what we do see here is, say, prior versus posterior predictions in one particular site, for example. You can see that the prior prediction, as you saw before with ensemble members, is quite wide, and the posterior is a lot narrower, but sort of focusing on where the data is. So what's behind this is a smaller region in parameter space that we know corresponds to these runs, right? And now we know better what are the values of these parameters going to be. And essentially, this performs an uncertainty reduction due to available data. This is the same result really shown slightly differently. As you saw before, these red curves are the ensemble members, and then the posterior is we are able to decompose the variance and kind of color code the error bar or this cloud of the prediction with various parts, including posterior standard deviation, which is essentially parametric uncertainty, but also accepting some surrogate error, which is in this case small, and model error, which typically is the one that is the largest contributor of the overall uncertainty. So the fact is that if we only use this side data, we would probably get very small model error. We definitely get very small model error, and we fit much better than what is shown here. But this is particular exercise where we use the whole Western Hemisphere data set or FLUXNET data set. And with that, when we do calibration, we need a single parameter that controls all sides. That's why extra model error component has to be built in, and that's what this result shows. This is the same type of result, kind of sliced differently on a specific time frame, May 2002. On prior on the left, and on the top is the bias, basically between model mean and data. On the bottom is the predictive standard deviation. This is on the prior side, okay? On the right, it's on the posterior side, where the both bias is reduced, as well as predictive standard deviation is reduced. So this really is the same type of result that I showed in previous slide, only sliced differently. And if you wish, the same thing over time resolution. So again, you can see kind of earth breathing here, and on the left it's prior, colors are more red. On the right, on the same time scale, we barely ever reach red because prediction uncertainty is reduced and bias is reduced as well, as you can see from top right plot. All right. So one other sort of exercise I wanted to highlight here is without model error implementation in the calibration process. This is the top plot. The FLNR, for example, the most important parameter, posterior, is all over the place for various sites. The site PFTs are on the x-axis. Basically, for each site to fit the data, FLNR goes out of its way with very small uncertainty and tries to fit. This is essential sign of overfitting. And but because we are assuming a single FLNR, this is the reason, because we are assuming single FLNR across all sites and all PFTs. If we think of it from model error perspective, we built in additional statistical component in the parameter with embedded model error approach. We do get a lot more stable result and a more fair representation of uncertainties on the parameter after the calibration. So as a summary, I'll mention that we constructed spatio-temporal surrogate to approximate ELM, Karhunen-Loève, which is principal moment analysis plus polynomial chaos expansion, and the surrogate is orders of magnitude cheaper than ELM. Global sensitivity is a free byproduct of it, really, and Bayesian calibration uses online evaluation of this surrogate with FLUXCOM data, and this extra piece model structural error component also built into Bayesian likelihood allows us to formulate predictive error bars that do not overfit as much as in a classical Bayesian calibration sense. So there's several steps we have next, but one of them is building a global calibration framework to construct ensembles with land biogeochemistry active, because the one we have here is a simpler case without that being active. So it will be higher expense, perhaps a smaller ensemble size. And determining sensitive parameters for variables that couple the Earth system, as well as engaging with ELM to prioritize datasets to be used to integrate with ELM. Here we only use FLUXCOM and we only use GPP data. So that will help us find best parameters and best parameter regions to use in future offline and coupled experiments. I think this is all we had, and we're happy to entertain questions in the last 3 or 4 minutes. Thank you.